What's up, baby bio penguins? It is Friday. It's Friday. Have an FRQ Friday. Yeah, I still don't know why I do this every week. So, hi, welcome. Guess what? 3 weeks. Yes, we have 3 more weeks until the first administration, which means that 3 weeks from today, you will be done with your exam and you will be rocking out of there, right? So before we get started, I want to go ahead and just make sure we have just a quick moment of, y'all, you've got this. I believe in you. And I know you can do everything that you've set your hearts to, right? So remember that when you come, hi, Brayden. Remember that when you go to take that test, it is only that. It is a test. It does not define you. It does not change anything that's happened this year. You know your biology, and a test isn't going to say whether you do or don't know that biology, right? And if you happen to have to take this again in college, it's not the end of the world, right? Because you're having a great foundation. But of course, we don't need to worry about that because you're all going to make a 3 or higher because you're penguins and you're ready for success. So let's go ahead and get started with our FRQ Friday. So there are 3 questions that we're going to do today. So 3 we're going to do, we're going to do 2018 number 3, 2018 number 7, and then 2016 number 5. And so you can tell by those numbers that we actually have all short free responses today. And this is based on the old exam where there actually were 8 questions if you're wondering why there's a number 7. And so somebody also said, well, Ms. Jones, I'm having trouble with plasma mapping. So I said that I would do it at the end of the FRQ today. So after those 3 FRQs, we'll go through a quick example on how to do plasma mapping. And we'll talk a little bit about gel electrophoresis just because it was something that was hard for me to explain on the lives. I mean, on the stories. So I figured I'd go ahead and just do it in the FRQ today. So first question we have is, they're all ecology questions, but seagrasses are aquatic plants that reproduce sexually, which means that you need to have a sperm and an egg, right? A male and a female component. So the male seagrass flowers produce sticky pollen that is, you're welcome, Shy, that is going to circulate water from the female to the female flowers, which results in fertilization. A researcher claims that mobile aquatic vertebrates can also transfer that pollen from the male to the female in the absence of circulating water. So they're saying that if there is no water present, no water that's circulating, okay, that there can be invertebrates that can do that job of taking the pollen from the male to the female. So to investigate the claim, the researchers set up aquariums to model the possible interaction between the invertebrates and the seagrasses. So it says to use these symbols. So we have a square, a circle, and then like an oval with some lines on it. Use these symbols and the template aquariums to demonstrate the experimental design for testing the researchers' claim that mobile aquatic invertebrates can pollinate seagrass in the absence of circulating water. Draw the appropriate symbols in the negative control chamber and in the experimental aquarium. Do not use any symbol more than once in the same aquarium. So basically you're going to have a maximum of 3 things in each of these, right? You'll have a male, a female, and an invertebrate, or a male, female, and an invertebrate, right? So we first have to think for a second. What am I supposed to be doing? I'm supposed to be drawing symbols, right? And as we've said before, we're just drawing something, right? So can you guess on this? If you have absolutely no clue in the world, could you at least draw some of them in either of those squares? Yes, you can guess. So if you have absolutely no clue, you should guess on this, right? Draw whatever you think in each of those. Remember that you don't repeating any of those circles, right? Any of those symbols. So you would have either a square, a circle, and an oval, or you can maybe do a square and a circle. You can do a square and an oval, whatever this you come up with, right? So let's think about what the real answer is, right? So if I'm doing an experiment and I wanted to test something, right? I need to have the same thing in both of my chambers, right? So in order for me to test pollination, I need to have both the male and the female in both of my chambers, right? Because the male is going to have the pollen and then it needs to be transferred to the female for fertilization, right? And same thing here. We have to have the pollen to transfer the female for fertilization. The only difference is now I need to test that claim in my experimental. The claim was that there could be mobile invertebrates. So the only one that I'm going to put those invertebrates in is going to be my experimental aquarium because I want to see, okay, if there are invertebrates, can it transfer the pollen? If there's not invertebrates, does it not transfer the pollen? And that's what I'm testing here, right? I'm trying to figure out whether the claim was correct that those can help, okay? So there was a point right there if you had drawn those in that. They didn't have to be in that order, but just draw them in those little chambers. And that's an example of what you might see for question number 5. Remember, question 5, you're going to draw some type of diagram. You're going to, draw something. So looking here, we have to identify the dependent variable in the experiment and then predict the experimental results that would support the researcher claim that mobile aquatic invertebrates can also transfer pollen from male to female female flowers in the absence of circulating water. Okay, so real quick, what is a dependent variable? Well, dependent variable depends on the independent variable, right? It's going to be what is the result of the experiment, right? So if my independent variable was the presence of the invertebrates, right? So if I have the vertebrate present, what would happen? Well, I need to see, does the flower get pollinated, right? Does the pollen go from the male to the female component? Okay, that is what I'm testing. So my dependent variable needs to be that pollination. Does it get pollinated? And then if we know anything about pollination, does it result in the seed? Okay, so am I able to transfer pollen from the male to the female? That would be my dependent variable. Well, how would I know whether that happened? Hey, we're doing FRQ 2018 number 3. So, if we're trying to figure out what kind of results I should get, I should be actually able to see that they were transferred, right? So I could talk about pollination, right? And my prediction would have been that more pollen grains would be transferred in the experimental aquarium. I could also talk about fertilization, in which more of the plants are going to be fertilized within my experimental aquarium. Or I could talk about just reproduction, about the production of that seed. And then I could say that more seeds were produced in the experimental aquarium. The whole thing that we're seeing here is if my dependent variable, right? So if we were able to transfer the pollen to the flower, to the flower, okay, will I be able to pollinate, fertilize, or reproduce? Okay, those were all the things that could have happened from that pollen being transferred. And then your prediction is just saying, what would I have found in the experimental that showed that? Okay, so not too difficult of a question. And there was a lot of parts where you could have just guessed and still gotten some points, right? You could have just randomly guessed the pictures, right? You could have just randomly made the prediction. So there's a lot of places where you just need to try, right? Don't ever leave anything blank on the test. And I think it was Michael Jordan that said, you miss 100% of the shots you don't take, right? You have to at least take the shot. You have to at least try. No, not on this one. This one was a short FRQ, is cut and dry, right to the point. So there really wasn't much else you had to say. You could have just been straight to that point. And a lot of times students, and I'm the same way. I'm long-winded. If you haven't figured this out for my videos by now, I don't know where you've been. Some of you have to say a lot. For me, I have to write a whole paragraph, and then finally at the end of that paragraph, I get the point, right? I know my content. I know exactly what I should be saying, but for me, as I'm writing my FRQ, I'm thinking through it. So I'm making my justification before I'm making my predictions. I'm explaining because I want them to understand that I know what I'm talking about. And that's just me as a writer. I've had students who could just write one sentence and get all the points with no problem. So it's all depending on your writing style. And after basically an entire year of writing FRQs, you've figured out your writing style, and you know how to get those points. But if not, this is what we're doing these for, is so that we can kind of see the trends and stuff that we might find from these FRQs. So our next FRQ is 2018 number 7. Now, this is a genetics question that connects into natural selection. I have no clue why I put this here, but it's okay. So I'm going to tell you right now, before we look at this question, this is one of those that's going to throw you off, right? As you read the question, you're going to go, what? What is she talking about? And that's what happens on the AP exam. You're going to read through a question and go, what is this even talking about? Make yourself little notes on the side, jot yourself down, circle things, underline things, make it so that you understand what you're reading, right? Because this is a unique, special situation that they have plucked out of nowhere's land to say, okay, hey, Melanie, to say, hey, let's make sure they know their biology, okay? So it's unique. Just go with that, okay? So in the tongue solefish, sex is determined by a combination of genetics and environmental temperature. Yeah, sometimes the sex is determined by the temperature. A lot of like alligators and whatnot, they're temperature selected. I think turtles are the same way. Genetically male fish have 2 Z's. So unlike humans, the males in this are going to have the same chromosomes. So we have 2 Z's. The genetically female fish have 1 Z and 1 W chromosome. So they're ZW. When a fish is raised at 22 degrees Celsius, ZZ develop into phenotypic males and ZW develop into phenotypic females. Okay. However, when the fish are raised at 28 degrees Celsius, the Z is modified as Z star. Z star W individuals develop as phenotypic males that are fertile and can pass on the Z star to their offspring. Even when the offspring are raised at 22 degrees Celsius, a cross between ZW and female are shown in the Punnett square. So we cross a Z star Z and a ZW. What? Did you say something about Z's? You said Z's W? What are you talking about? That's exactly what I did when I read this question, actually the year that it was assigned, okay? So exactly what I just said. You need to draw yourself out something to help you, right? So as I read through the prompt, I wrote them stuff down. Okay, so at 22 degrees, if I have a ZZ, that's male, a ZW is female, okay? If I have a Z star Z, that's male, and a Z star W, that's male. Okay, cool. But at 28 degrees, if I have anything with a Z star, exactly, Andrew. If I see anything with Z star, it's going to be male, okay? So what's happening here? So we did our Punnett square, right? And so if I look here, I have a ZZ. Sweet. That is a male. Easy peasy, right? And I have a Z star Z. So that is a male, right? I have a ZW. So that's a female. And then I have a Z star W, which is also a male. So it said to predict the phenotypic males among the F1. So they gave me the F1 cross of the cross shown in Punnett square if they were raised at 22 degrees. So using this 22 degrees chart that I made, I was able to fill this in. So you would see that 75% of them are going to be males. I will tell you that many students got this wrong because they could not understand what the prompt was talking about. Okay. So you want to make sure that you can get yourself a key if you need to help yourself to quickly look back at it. So you don't have to keep rereading that prompt, right? The more times you have to reread that prompt, it's going to be harder for you because you have to keep, you're losing so much time, right? So the second part says at least one Z or Z star chromosome is necessary for the survival of fish. A researcher crossed two fish and observed a 2-to-1 ratio of males to females among the offspring. Based on the information, identified the genotype of the male parent in the cross and ascribed one fitness cost to the female by mating with this particular male. Okay, I have a 2-to-1 ratio. I thought there were 4 things that came out of the Punnett square. Oh gosh, what am I going to do? Okay, we can do this. I believe in this. We can do this. Okay, so if I fill in my Punnett square, I got a male. And I got another male. Okay, so I have my 2 males, right? And then I have 1 female. Cool. What's happening here? Okay, so if I don't have anything there, that means that it didn't survive, right? So that means that it must have died, right? It did not survive, right? Because I have to have a Z or a Z star in order to survive. So that must mean that there's no Z or Z star here, right? So it must be 2 W's. That's the only plausible way to do this, right? Because it did not survive because it doesn't have a Z. Okay, so now let's work backwards, right? So if there's a 2W here, that means that that one must have been a W, and that one must be a W, right? I must have two W's that were crossed that gave me this. Okay, cool. Okay, so now in order to get this to be a male with a W, I have to have a Z star. Okay, so Z star. So here I have a Z star W, and then this, in order to get me a male here and a female there, there's a W and a Z. Okay, this must be a Z. Okay, so I was able to get my 2 males to 1 female, knowing that this one didn't survive, and I was able to see that I crossed a female with a Z star male. Okay, so that would tell me that my male must be Z star W. Okay, and then they want to say, well, what is a fitness cost? Okay, so fitness is the ability to pass on your traits to the next generation, right? So if you have any of your individuals, your offspring that do not survive, that decreases your fitness, right? So were all 4 of these offspring, were they able to survive? Well, no. We have 1/4 of our offspring passing away or don't survive, right? So that is a cost to my fitness, right? I'm unable to have an offspring between this cross, right? Because of the fact that it passes on as 2 ZWs, right? So that is the cost that we're looking for. This is where they're describing the problem, right? So the things you could have said is that you have fewer offspring that survive. You could say that 1/4 of the offspring were predicted to die. Or you could just say that some of her offspring will have the Z star chromosome and all of her males will have a Z star. So that also can be a problem, right? Because if this Z star keeps getting passed on, then that can also decrease the fitness of the future generations, okay? So again, if you have questions, you're more than welcome to drop them down in the chat. I can see the chat as we're going. Unlike the Q&As when I don't see the chat because I'm kind of working back and forth with Melanie. So you're more than welcome to, if you have questions, just put them down in the chat as we go. So our last question we have. This one has to do with interactions, ecological interactions, okay? So the graph above, which will end up being below in a second, shows the mass of plants from two separate species over time. The plants grew while attached to each other. So here we see them attached to each other. Here's species 1 and species 2, and they're growing, right? The plants were separated at the time, indicated by the vertical line. So this is vertical line in which they were separated from each other, and we want to see what's going to happen, right? So using the template, graph the predicted shape of the plant mass lines after separation of two plants if the plants were obligate mutualistic relationship. Okay, so if we have an obligate mutualism, okay, so let's first think about it. What is mutualism? Okay, well, mutualism means that they both benefit, right? So if they're both benefiting, I should see an increase, right? But here's the problem. It says that they're obligate, right? So whenever you see obligate, you know they're obligated to be together. They cannot survive without the other. It makes you think about the the termite and the protozoa? I think it's a protozoa that lives in their intestines. And the protozoa's job is to break down the wood for the termite, right? Because termites eat wood, right? So the protozoa breaks down that wood and gets the cellulose broken down, right? Releasing the glucose. So the termite gets food. And then also the protozoa gets food. So that would be an obligate mutualism. If you were to kill off that protozoa, your termite could not survive and it would die because it can't break down the cellulose, right? So when we talk about obligate mutualism, they're obligated to stay together. And so with that dotted line, I'm separating them, right? I pulled these two apart. And so by pulling them apart, that's going to cause both of these lines to decrease. They're not going to have as much plant mass because of the fact that they are obligated to stay together. They cannot live without the other. They are dependent on each other. And without that relationship, both of these individuals are harmed. So hopefully that makes sense, right? So because they're obligated to be together, you're going to see a decrease in plant mass because of the fact that separating them inhibits both of them, right? Because they had to be together. The second one we see, it says on template 2, graph the predicted shape of the plant mass lines if species 2 plant was a parasite of species 1. Okay, so parasite, and if we're talking about parasitic relationship, right? A parasite is going to be in which you have one that is harmed and one that is benefited, right? So your parasite is going to benefit and your host is going to be harmed, right? So plant 1 is my host and plant 2 is my parasite. Okay, so if I separate them from each other, what will I expect to happen? Well, plant 1 no longer has a parasite, so it's going to be able to grow, right? Versus plant 2 is the parasite and no longer has a host, so it's going to decrease. So I should see it kind of spreading apart like this, okay? So species 1 should continue to increase while species 2 should level off or decrease, okay? And the reasoning why is because the parasite requires an association with the host in order to survive because it's going to harm the host. And then without that relationship, the parasite cannot survive without the host, okay? Because we're looking at a parasitic relationship, the parasite has to get the materials from the host and it's going to harm that host in the process. So by separating them, host is going to increase and the parasite is going to decrease. So hopefully that makes sense. So that was our 3 FRQs. Remember, I said that we're going to go over plasmid mapping, but before we go on to that, I want to remind you that next FRQ Friday is going to be over these 3 questions. So 2018 number 5, 2017 number 4, and then 2016 number 3. I'll meet y'all again next week on the 30th. So plasmid mapping, because someone asked me to go over this, so I figured, hey, let's do it. So this is a super old question, like super old. I think it's like 2007, I think, or so. So plasmid map is 100 kilobases in length. The plasmid DNA was digested complete with 2 restriction enzymes in 3 separate treatments. There's EcoRI, HAE3, EcoRI plus HAE3, which is a double digest. The fragments are then separated by gel electrophoresis as shown. So you can see the product from the gel electrophoresis. And so when we look at this, I can see a lot of different things, right? So there's a molecular weight standard that shows you the length of each of the fragments, right? So if something is this long, it's 10. If something is this long, it's 60. If something's this long, it's 90. So you can kind of get the scaling right there, okay? So if I look here, I see that Echo has 2 fragments. Hay has 2 fragments, and then if I use a double digest, there's 4 fragments, right? So let's think about this, right? Oh, I forgot to ask the question. Using the circle provided, construct a label diagram for the restriction map. No, don't pause. Using the circle provided, construct a label diagram of the restriction map of the plasmid. Explain how you developed your map, okay? So all I'm working on here is how we make that map. I'm not going any more than that, okay? So if I look here, I can see that Echo RI has 1 fragment that's 70 and then 1 fragment that's 30. So I write that down. Hay 3 has 2 fragments, one that's 60 and one that's 40. So fill that in. And if I use Echo and Hay, I have a 40, a 30, a 20, and a 10. So fill that in. Okay, now let's think for a second, right? Oops, sorry, y'all. I have a circle. My circle is my plasmid, right? Because we know plasmids are circular DNA, okay? If you cut your circle once, it just makes 1 long strand, right? If you cut it twice, you now have 2 fragments, okay? So I need to figure out, well, how many times did my restriction enzyme that's going to cut my DNA, how many times did it cut, okay? So if I look here, I see that there's 2 fragments. So my Echo RI must have 2 cuts. My Hay must have 2 cuts. And then my double digest has 4 cuts. Basically, it's the 2 cuts from the Echo and 2 cuts for the Hay. Okay, so it should have 4 cuts because there's 4 fragments. So how am I going to do this? Okay, so you have to pick one or the other. For me, I just picked the first one because it's just easiest for my brain. So since I know there's going to be 2 cuts, I make 2 lines on my circle to say, okay, here is where Echo is going to be. I make an E to symbolize that's the Echo, and I make another E on this one to symbolize it's the Echo. Okay, now I'm going to arbitrarily put these numbers down. I put a 70, I put a 30, nothing too crazy, okay? With a lot of this is going to be a big trial and error, right? So I put those cuts down. The next step is you now need to jump down to the step 4, which is this last line, okay? I have 1 fragment that's 30 and 1 fragment that's 70. How can I make a 40, 30, 20, and 10 by cutting it? So if I cut my 30, I can make a 20 and a 10, right? If I cut my 70, I can make a 40 and a 30, okay? So now we need to say, okay, well, is this right? Okay, so I should have, if I go from Hay to Hay, so if I go from H to H, I should have either a 60 or a 40. So 40 plus 10 equals 50, which is not right. Okay, so what did I do wrong? Let me think for a second. Okay, what about if I switch these two, right? Because this whole thing right here had to be 70, and I just arbitrarily put the 40 here and the 30 there, right? So what happens if I just switch them? So if I switch them and I go from Hay to Hay, I now have that 40. If I go from Hay to Hay, I now have that 60. Let's double check, make sure we're still good on the echoes. Echo to echo is 70. Echo to echo is 30. I have a 10, a 20, a 30, and a 40, and I am done. So, hope that was helpful. Don't forget to download your app for your AP exam. Good luck to everybody as they're studying for the AP exam. Remember, we have 3 more weeks until the first administration, that first paper administration. And then we will have the second administration of digital, which will be on May 27th. And then your third administration, which is also digital, will be on June 11th. Remember, you've got this. I believe in you. You're an AP Bio Penguin. You're dressed for success. Have a good day. Bye.